Peter Steinberg from the University of New South Wales in Sydney, Australia, although it doesn't sound like he's coming from Australia when he speaks. So thank you for traveling this distance as well. His talk is on seaweed-bacterial interactions, disease, climate change, and bacterial communities. 30 minutes, I'll stand up and see if I'm...
I'm from Brooklyn, right? So like the other speakers, I want to thank Mark very much for inviting me to participate, and also Tanya and the rest of the team, and to the Teasleys for making all this possible. It also occurs to me that I'd like to thank Mark very much for another reason, which is that after 30 years, he has actually sat down and stopped talking on my behalf. Don't get used to it. So, as Tanya said, I'd like to talk about seaweed-bacterial interactions in the context of climate change and disease. And before I jump into this, I want to set some boundaries. So it's not really clear why I'm here, because I don't really work on corals. And in fact, this is my only coral slide that I will share with you today. So hopefully this is something of an interlude, a temperate algal system interlude. Having said that, I've been very interested to hear what people have said, because I am starting to work on corals in Singapore. So why am I here? I think I'm here to fly the algal bacterial flag. So while, as I've listened to the talks here, at least 4 key groups of organisms have emerged as the players in the kinds of systems that we're talking about. The corals themselves, of course, herbivores, particularly herbivorous fishes, and then seaweeds and bacteria. And this, by the way, is what real seaweeds look like. So while I don't work on the 2 on the right-hand side, I have worked for a number of years on seaweed-bacterial interactions. And so hopefully some of the things that we've learned from these temperate kelp and other seaweed communities and their interaction with bacteria can lend some insight into the kinds of things that people have been discussing here. So I really want to talk about, well, 2 things, but 2 emergent things and 2 more specific things that will have to do with some of the systems we study. So hopefully the emergent messages that arise from my talk are that, I think we've reached a stage where we really have to acknowledge that microbial effects and disease are persistent ecological factors in the same way that all of us have studied competition, predation, disturbance, and what have you for many years. And, from the talks that have happened already today, perhaps that's not news to this crowd, but I think that in general, with respect to the kind of ecology that we talk about, aside from the recognition that there are occasional catastrophes that seem to be driven by pathogens, I don't think we yet treat disease and microbial impacts in the same way that we treat other ecological factors. And certainly in terms of evolutionary interactions, the ones with microbes are by far the most ancient. The other thing that I want to talk about, which is related to that, is because of the development of some of the new molecular tools and other techniques in microbial ecology, I think it's time that we can start to move towards the unification of microbial and eukaryotic ecology, which for pretty much the history, professional history, of most of the people in this room have proceeded as largely completely separate disciplines. So it's time not to do that. So hopefully those things will emerge in the context of 2 specific questions or 2 specific systems that I want to talk about. One of that is, what are the mechanisms and impacts of seaweed disease? And how does that interact, excuse me, with environmental change? And by environmental change, I mean things like ocean warming, climate change, but also urbanization and other anthropogenic effects, although I won't talk so much about that today. And then finally, I'd like to try and take some of the things, some of the concepts that we've learned from eukaryotic ecology and ask questions about the community ecology of surface-associated bacteria. So what's going on in those communities? How do they interact? What are the dynamics? And in fact, is that they cascade back to effects of those. Okay. So let's see if we can do all that. So one of the high-profile predictions of environmental change, particularly climate change, is that there'll be an increase in the intensity and frequency of disease. People have argued that both in terrestrial and marine systems, and we see some really quite striking examples in things like seagrass beds, as well as some well-known coral examples where people like Eugene Rosenberg have identified particular pathogens and the like. Australia, Southeast Australia, is a wonderful place to work on climate change and ocean warming because it's a global hotspot. Pixar got it right, basically, for those of you who saw Finding Nemo. So we have a tropically dry, southward flowing East Australian current, which is getting stronger as the decades roll on. And one of the effects is it brings tropical fish to Sydney Harbour and further south. Those fish are now starting to overwinter. So in fact, what we're seeing is the tropicalization, if that's a word, of the temperate coast of Southeast Australia. So the question is, what kind of impacts is that having on those communities there? So we worked for a number of years on this red alga, Delisea pulchra, which is arguably the most common foliose red alga in that part of the world. So as you can see, it forms reasonably extensive beds on the sea bottom. We've done a bunch of things with chemical defenses. Over the last few years, though, we noticed what we're calling a bleaching disease. So it's not big news to people who study seaweeds, that seaweeds occasionally bleach, but the standard photo bleaching has a very typical phenology in which it's bleached up top where the irradiance is the highest, and it gradually moves towards the bottom of the thallus. So we don't see that for this particular phenomenon. We see bleaching at the bottom or the mid-thallus. And it typically starts as a small point and then seems to spread out, looking very much like an infection. So that was the hypothesis. The hypothesis was that this bleaching phenotype was in fact a bacterial infection or a microbial infection. It has significant consequences to the plants, and I'll talk about that later, and it's also very common. So this is at a couple sites near Sydney and over a few years. And in these populations, at some times, 80 or 90% of the thalli show evidence of this bleaching phenotype. And as you see, it typically happens in the warmer, this is December and January, it's warm in Australia. So if we have questions about, is this a bacterial effect? So one of the things you want to do first is start to characterize the microbial communities on healthy versus supposedly diseased plants. And we've done this, using standard techniques by sequencing the most commonly used taxonomic marker gene, 16S ribosomal RNA. And if you do that, Katie showed similar sorts of data, you show, perhaps not surprisingly, that you get a big difference in species richness between healthy thalli and bleached thalli. And as Katie also showed, the diversity of bacteria in these things is quite high. The one thing that's interesting, though, is, in fact, we see the richness, species numbers, of bacteria on visually healthy tissue next to bleached tissue on the same plant. We see those communities, those microbial communities, grouping with these damaged tissue. So what we're seeing is we're seeing systemic effect on the entire thallus before we actually see any obvious phenotypic effect. And while that's not definitive evidence of issues around causality, at least it suggests that something's happening with the bacterial community before the bleaching event occurs. Well, something is happening. It's a similar story for bacterial community structure. So if you then assign taxonomic status, they say operational taxonomic units in the world of bacteria, to those sequences, then you can show a very similar thing. You show that bleached and adjacent tissue, that adjacent tissue is a tissue next to bleached, but it's unbleached, group separately from healthy plants. So again, there's a systemic change in bacteria that are on the whole thallus. And it turns out that it's a relatively few taxa that are changing quite dramatically. So for a number of years, excuse me, I'm losing my voice here. So we worked on the chemical defense of Delisea pulchra for more years than I like to think about, actually. And we characterized the compounds, and we looked compounds on the surface. And we also know that they have very specific effects against bacteria. So they interfere with something called microbial or bacterial form sensing, which is a way that bacteria talk to each other using small chemical signals. And they were the first known natural inhibitors of form sensing. So there was a known antibacterial effect, so it was not surprising that we asked the question, are these compounds active against microbes, and do they play a role in preventing this bleaching phenomenon? So again, we see, like with the microbial communities, we see big differences in the levels of Furanones in bleach versus healthy tissue. And they have, like the microbial communities, already begun to decrease in the tissue that is as yet unbleached. And they're also consistent with the temporal pattern that we see, lower in summer than in winter. So we'd like to then test this hypothesis which arises, which is that something is happening to these Furanones, which, following their decrease, they get colonized by bacteria, leading to bleaching. So we do this trick with Delisea. The compounds are brominated, so all the halogenated metabolites that it makes are brominated. So we do this trick where we grow in the lab, we grow baby Delisea in the presence or absence of bromine. And in fact, that trick's been known for a number of years, but we took it and applied it in the context of chemical defense. You know, we've done a variety of things to try and convince ourselves and others that this manipulation has no impact on the plants. We've added higher levels of bromine than we normally add. We've taken it away. We've done it for plants, for algae that don't make brominated metabolites. We've done it for some other species. As far as we can tell, it has no whole thallus effects or specific ultrastructural effects, other than effect on the gland cells that make the Furanones. So if you then take these baby plants that now are with versus without Furanones, and you outplant them into the field, and you put them in small screen meshes so that only bacteria can pass back and forth, you can test the effects of Furanones on this putative bacterial disease and test the effects on bleaching. So this is that experiment. And we did it in a couple of years. And so here are the Furanone minus plants, and here are the Furanone plus plants, and this is what we did. So if you take away the Furanones, then the bacteria which get in through those meshes cause bleaching in hours. We did these experiments for 24 hours. Okay? So I think starting to speak now towards the causation of this disease. The thing that is not so easy to do in the field is to look at the kind of stressors that we have hypothesized cause stress the plant or affect the bacteria and lead to either a decrease in Furanones or increase in virulence and thus the bleaching disease. So not surprisingly, given that bleaching mostly occurs in summer, there's a strong correlation, highly variable, but a strong correlation between temperature and the incidence of bleaching. And the correlation actually between water temperature is much stronger and bleaching is much stronger than for some other obvious environmental variables like light. So what we did to initially address the effects of temperature on the system excuse me, is to take it back into the lab and do a series of inoculation experiments where we cultured up bacteria from the surface of Delisea and then added them back to either bromine Furanone minus or Furanone plus plants at two temperatures. One towards the lower range that these plants see in the field in summer, and one towards the higher range. If you do that, you see two basic effects. This is with one particular pathogen. So in the presence of Furanones, at least for this particular strain, you see no effect on these plants. At 20 degrees, in the absence of Furanones, and you inoculate these plants with this pathogen, then you see the formation of a biofilm on the surface. At higher temperatures, you see a biofilm and you see invasion. And so in fact, we've used a green fluorescent protein tagged version of this pathogen in these experiments. And you can actually watch by video. Rebecca Case, the woman who led this work, has this lovely little video of these things invading, getting into the cells, and causing bleaching. So we are also starting to find now, as we look at other strains of pathogens, that we're starting to see some effects in our high-temperature Furanone plus algae as well. So one of the issues, it's not entirely surprising if the hypothesis is that it's driving down, temperature is driving down Furanones as well as enhancing virulence. But the small scale of these experiments makes it a little bit challenging to nail that down in these things. So we're in the midst of moving some of these inoculation experiments into the field. And we certainly can induce bleaching in field plants by doing those kinds of things, but we're kind of right in the midst of this and don't quite have that part of the story together yet. So the final piece I just want to add to this is, so we can show this phenotypic change as a function of chemical defenses of bacteria. But the question is, what effect does it have on the allergen? And so typically when people think about disease, they think about these very broad-scale catastrophic effects, which I think is partially because that's when you notice it. So this bleaching phenomenon has little or no effect on survival of individual thalli. But in fact, what it does, it essentially neuters Delisea. So Delisea, foliose red alga, produces its sorile, its reproductive parts, on the tips, the distal tips. The bleaching typically occurs mid-thallus or lower. And as that tissue bleaches, the thalli lose their upper half, typically sometimes more. So in fact, what they do is they lose the capacity to reproduce for the most part. Some residual is left here. So it's a sublethal, but certainly very significant effect on the fitness of these populations. And one of the things we're just starting to explore is whether or not this is also affecting the genetic structure of these populations. Bleaching, perhaps not surprisingly, also increases excuse me, susceptibility to herbivores. So the model that's kind of evolved here is a model of increasing stress on the plant, which leads to a decrease in chemical defenses. It also potentially leads to an increase in virulence of the pathogens, because it's well known, at least for human pathogens, that virulence factors are temperature sensitive. This then leads to this bleaching phenomenon, which then has consequences to the fitness of the plant. And one of the comments I made earlier on is, you know, these things are exposed every minute of their existence to 1 million bacteria in every milliliter of seawater. It's this constant, intense, in my view, selection pressure that's been there for the entire evolutionary history of this algae. So I think there are hundreds, thousands of potential pathogens here. I mean, we just started screening things without looking for any bugs in particular, and we got repeated hits. So I think these things are surrounded by opportunistic pathogens that, in light of some of the environmental changes that these systems are facing, will be increasingly a factor in the ecology of these algae systems. Oh, and by the way, we're doing other things as well. We're starting to look at some of the major habitat-forming species, kelps, and a big fucoid that occurs there. And, you know, we're seeing, if you look, similar kinds of phenotypes that may be bacterially caused. All right. Somebody else had a soapbox moment earlier, I can't remember who. This is my soapbox moment, which I've probably already done, which is that, you know, arguably the greatest diversity, greatest biomass, greatest everything of living organisms on the planet are microbes. If you took away coral, seaweeds, and fish, the planet would continue very happily. If you took away microbes, pretty much life would cease almost instantly. So if we as ecologists are really going to be serious about trying to understand general principles of ecology and environmental science, you can't ignore environmental microbiology. And I say that, I think safely, because I'm not a microbiologist trained. So this is all born-again evangelical stuff. Okay, so two things, and what I've tried to do here is present some of the work that we're doing that draws on some of the concepts of eukaryotic ecology and then feeds back. And, you know, time is short, so it's going to be real snapshots. So we have looked at succession microbial communities on Delisea pulchra, trying to understand what's the pattern of recovery of microbial communities after disturbance, and what are the mechanisms governing the succession. And here, it may just be because I'm old, but it's also because I have enormous respect for a paper by Connell and Slatcher years ago about mechanisms of succession. But also, once you start looking at the dynamics of the algae, the bacteria in the algae, does it actually have consequences to some of these larger effects that I've been talking about? So what we did was do what we consider to be analogous experiments to what people do in succession in any community. You try and start with a fresh habitat. So we did a number of things, including antibiotics, to try and denude the surface of the Delisea microbial communities. And then we put them back out in the field and measured colonization over time, tried to characterize those bacteria, and compared that to a variety of other controls in inanimate surfaces and seawater and what have you. These are MDS plots where you can show the trajectories of these microbial communities over time. So we did this initial work embarrassingly a long time ago. And so we used a molecular fingerprinting method as opposed to doing a straight sequencing one. But in general, what you see is you see, after about 12 days, you see a return to those defaunated communities back to where they started and back to what the communities on unmanipulated fetal plants look like. There are some outliers, there are some instances where it looks like the manipulated plants haven't quite gotten to the controls. But in general, there seems to be this predictable successional pattern back to a Delisea community that we define. So the question was, are there kinds of mechanisms underlying these kinds of phenomena that people have described for forests or for fouling communities or the like? And there's a relatively complex sequence of events and methods that we use to try and test that question. But essentially, because we were measuring Delisea at different stages in the successional process, we could characterize early successional and late successional species. So we isolated, cultured some of those up, and inoculated both Furanone plus and Furanone minus plants, and looked at the resulting communities. And you do it in ways where you can mix and match early successional and late successional species. So you can test some of these notions about successional mechanisms by inoculating the appropriate bugs at the appropriate time. So the results are kind of interesting. This is one such experiment, and it's a little bit complicated, but the key thing here is that the early successional species are in orange, or the early successional species, singular, is in orange, the late successional in red. Now these are pared back experiments in the sense that we're working with a couple of microbial species, we know the diversity is high. But within that constraint, what it basically shows is that Furanones are strongly inhibitory, which is no surprise, both early successional and late successional. But if you remove the Furanones, then you see inhibition of late successional species by this early successional species. And then if you don't inoculate with early successional species, but inoculate initially with late successional species, you get the release of that, and you get a great increase in bleaching of the thallus. So not only are the Furanones acting as a mechanism to prevent microbial attack, but depending on the sequence of colonization of the bacteria, it also appears to have an effect on consequences to the plant. So I would argue, I guess, that those internal, micro-scale community dynamics within the microbial community of the plant also have an effect on subsequent infection, colonization, and the like. And what it really does speak to, in my view, is this notion that was coined, I think, by Forrest, of the whole of violence. Tell me that he came up with that term here. Yeah. So he's not here, so I would do the Laban thing to him if he was here. But this notion that seaweeds or corals or higher organisms are not just seaweeds or corals. They're the totality of their microbial community and the host. Okay, so the last example I want to give is that work on Delisea got us to a place, you know, sort of tight, almost deterministic control of community development on the outer surfaces. Now, I'm not a particularly deterministic guy, so that troubled me. You know, it kind of went against my overall philosophy of life. So when you get results that do that, what do? Well, you shift systems, right? So we've looked at another seaweed, and that's the thing that occurs everywhere, well, in our case, old Australians, but all of it is everywhere. You know, abundant, blooms. And we've looked at the microbial communities on Ulva. And we've done this in a snapshot kind of way. So we compared communities on Ulva, now using 16S sequencing, with communities in adjacent seawater. And this is just clustering of those community structures based on 16S sequences. And two things become evident. The first is that the bugs in seawater are very different than the bugs in Ulva. This is not a big surprise, although, you know, those bacteria in Ulva have to be coming from somewhere, presumably in seawater. But the variation between individual Ulva communities is enormous. And so of the 800 or 1,000 phylotypes that we identified from our different samples of Ulva, there were only 5 that occurred on each of our Ulva samples. And so those communities differed by 80% in many cases. So that speaks to something other than the highly deterministic bacterial community. So we wondered what was going on here. So this is an excellent slide. But it makes a simple point, and so the detail is, of course, not critical here. As well as using sequences to identify taxa, you can also use them to identify categories of functional genes, and I think we heard some of that from Katie earlier on. And there are large databases that allow you to compare your sequences to what's in the literature and conclude things about at least categories of functional genes. So if you look then at this functional gene level and ask questions about, well, what's more common on Ulva versus in seawater, then you get a variety of things falling out. If you then cluster those functional genes as opposed to the taxonomic markers, in fact, you get very tight clustering. And you get much stronger, much greater overlap between the different communities on Ulva as a function of those functional genes, than you do using phylogeny or taxonomy. Those functions do not map onto phylogeny. So again, the only point of this phylogenetic tree, another tough slide, is that these colored pie charts, which represent the presence of different classes of functional genes, occur throughout the entire phylogenetic tree of bacteria. So this is the whole ball of wax, right? So there's no phylogenetic pattern to those functional genes. They can be packaged in anything. But to succeed on Ulva, you need at least some combination of those functional genes. So what does this sound like? So I think there are two messages from this. The first is, as opposed to this tight deterministic system that seemed to be the case for Delisea, in fact, this is a lottery model for community assembly. This was originally proposed in the 70s by people like Peter Sale and Peter Doherty. And it was proposed for coral reef fishes, so I finally come back full circle. And they basically argued that within a functional guild of reef fishes, that it didn't matter who eventually colonized the coral reef, as long as they had the functions that were associated with that guild. And so it was a lottery, whichever particular species got there first. So I think that's what's going on here, except I think the difference is that the guild is enormous. You know, these guys were talking about Chaetodontids. They're talking about specific families of reef fish. Here the guild includes members of all the bacteria. So we have this lottery hypothesis with an enormous number of members of that guild. So that's kind of interesting because you can kind of transfer this eukaryotic theory into the world of microbes. But I think there's perhaps a more emergent message. And where we got to with this is, in fact, we had to seriously question whether it made sense to talk about community assembly of bacteria using species. So in fact, what it looks like for this system is community assembly of genes. So as long as you're a bacterium that has the appropriate genes, and you can colonize that surface. And we're not the only people to start thinking about how there's this big disconnect between structure and function, between phylogeny and function, in a variety of, in fact, very different microbial communities. So for me, having been raised as a eukaryotic ecologist, the notion that the species concept is not particularly relevant to one of the fundamental features of ecology, community structure, community assembly, that's a significant thing, at least for me. And so I think it speaks to the issue that, not only can we use some of the concepts of eukaryotic ecology in these microbial pieces, but, I think it's starting to feed back. And I think we can start to ask the question whether the way that bacterial communities function, both within themselves and in terms of their interactions with eukaryotes, whether in fact they abide by the same rules that eukaryotes do. Okay, so actually that was part of the emergent message. So two things that I tried to argue by pulling out some of those examples. One is that I think we need to treat disease seriously. I think we're getting to the place where there are tools that we can address, disease as a persistent, recurrent ecological phenomenon, both in terms of the causative agents as well as some of the outcomes. They're hard. Some of the experiments are hard. The diversity is enormous. The time scales are very short. All that kind of stuff. And we've heard some of those issues in the interactions that we've had today. But it's possible. And then finally, I'd like to think that we as a community of ecologists can move towards the unification of microbial and eukaryotic ecology. Because as I say, if you ignore the greatest proportion of diversity on the planet while trying to arrive at general ecological principles, you're missing something pretty significant. I've lost the slide that had all the pictures of these people. So I'd like to thank all the folks who did work and the Australian Research Council, who were the primary funders of the things I talked about. Thanks.
We have time for questions if anyone has any questions for you.
Yeah, Julie.
So you showed that in your simplified early successional stage of microbes, that they could suppress the late successional stage, which is also simplified. And then you have these guilds that could be guilds of various successional stages. Based on what you know so far about the genetic community or the community of genes, do you think that within a successional stage that you have a majority or a balance of positive interaction between these microbes or negative interaction between these microbes, or whatever complement of positive and negative, that helps decide which microbe can co-occur with which other microbes at a given successional stage? And the reason I ask this is because the algae, the seaweeds, Delisea that you talked about today, and the ones that Mark and Doug talked about today, are among marine macroorganisms, seem to be unusual in that they actually seem to produce their own secondary metabolites. A lot of marine invertebrates don't produce secondary metabolites the way we used to think they do. It's the bacteria that are within their tissues that make the secondary metabolites. And I think that it's likely that those bacteria evolved to make those secondary metabolites because of interactions between them and other bacteria, not necessarily with the host. The host might just be the food and the home. So do you think that, what are the ecological interactions within a guild then?
So that's a long question. So let's see. So I would Well, I disagree with one of your propositions, which is that the algae which produce their own secondary metabolites are somehow unusual. I mean, they're the dominant, a lot of the seaweeds that we worked on are the dominant habitat-forming organisms in those systems. And to my knowledge, almost all the secondary metabolites that we talk about in the context of, seaweed chemical ecology are produced by the seaweed, not the bugs. So they may differ in some respects to invertebrates, but I don't see that as an exception. So I have two general comments to, I think, your question, because the simple answer is it's complex and I really don't know yet. But I think a couple things impinge on the answer. The first is, I think there's a structure-function issue that relates to, call it environmental stringency. So I think as you get to very stringent environments, and maybe Delisea is a very stringent environment because of the chemicals produced, I think you get more of a convergence in structure and function. And I mean, we know that in some very extreme environments. Extremophiles are particular subsets of bacteria, right? And they have particular functions. So I think part of the answer to how those interactions play out is how stringent is that environment. If it's something like Olva, then maybe all bets are off and anybody can come in and there's every possible combination. If you have a more stringent environment, then you start to delimit those functions a bit more specifically. And whether that's likely to be positive or negative is still a little bit hard to say. So that's one thing. It's a long answer to a long question. The other is that, on the one hand, I talked about these experiments where I manipulated huge bugs, and these other descriptive things where you can show that there are thousands or hundreds of species. And so, those two things are not yet well matched. And so to be able to manipulate a community, individual species by individual species, it's going to be a challenging thing. And so I think we need to come up with ways that we can start to manipulate subsets, which will start to be able to address your question. We know that there are bacteria on the surface of Olva that are both very inhibitory against other things, as well as those which are fundamental to the development of the natural morphology of the plant. So both are clearly going on.
So some of the systems, I think, like dog and monkey, are so dynamic, after 10 or 20 days, and say that the coral is gone when bleaching is done. So how do you pay attention to disease? How do you really look at disease patterns and dynamic?
I think it's one of the reasons, I'll kind of flip the question on its head, I think it's one of the reasons why we haven't paid attention to disease. It happens, boom, and it's gone. And unless you lose an entire kelp head, you don't notice. Right? You have to start looking. I think it's as simple as that. You have to start looking. And maybe like a lot of things, once you start looking, you start finding it.
Just to add to that, that was just one reason why we started paying attention to coral disease, because it has had a drastic impact. So. Yeah.
And, in coral disease, for me, it was at least in a place where, people talk about bleaching, white band or black band. In most cases, there's a bit of a disconnect between what's actually causing those things and the observed phenotype. No question, something's going on. What the heck is happening to get there? Yeah.
The functional genes that are scattered all over the phylogenetic tree of bacteria, is that from lateral gene transfer?
Yeah, that's an interesting question. So one of the things that we do know, well, we know a couple things. Lateral gene transfer, right? So for those of you who are not in this world, bacteria can take packets of genes and shove them into some other, often completely unrelated bacterium. It raises some real issues like, how do you maintain coherence of the species? And maybe that's why we don't see structure function, right? The second thing is that that phenomena is more common in biofilms. So biofilms facilitate lateral gene transfer. And, what have I talked about, maybe without actually saying it this whole talk? I've talked about biofilms on the surface of seaweeds. And so we know as a general principle that it's more common. Whether a lot of those functional genes that I talked about are the consequence of lateral gene transfer, a difficult thing to know at this stage. So, on the actual answer to your question, I'll just kind of bail.
questions?